Welcome to the Talking Ketamine podcast, the show where we explore the science behind this fascinating molecule. This is episode 35, Ketamine and Stroke Recovery.
Thanks for joining us. Today we're looking at a new preclinical study that asks a really important question.
Yeah.
Can ketamine protect the brain during a stroke?
And that's such a critical question because, when you think about treating a stroke, most of the focus, almost all of it, is on getting that blood flow back immediately.
Right. Things like TPA, maybe mechanical thrombectomy. Speed is everything in that initial phase.
Exactly. Deal with the blockage.
Absolutely. Because that initial ischemia, the lack of blood flow, it starves the brain tissue of oxygen, of glucose. It's devastating.
There's a but coming, isn't there?
There is. It turns out that a lot of the really significant long-term damage, the stuff that causes lasting neurological problems, widespread cell death, often happens after that blockage is cleared.
When the blood flow comes rushing back in.
Precisely. That's the paradox. You fix the initial problem, but the return of blood flow itself triggers a second wave of injury.
Okay, so what is this second wave? What's it called?
The researchers call it cerebral ischemia-Reperfusion injury. Just IR injury for short. And honestly, it's like this invisible enemy in stroke recovery.
Invisible because the main crisis seems over, but the damage is still happening?
Exactly. It's a major reason why people have ongoing deficits, even after successful clot removal. Current treatments don't really address this IR phase very well.
So why is that moment when the oxygenated blood comes back? Why is it so destructive? What's actually going on at the molecular level?
Well, it's basically a sudden, massive chemical imbalance. The return of oxygen to those cells that were just starved, it kicks off something called severe oxidative stress.
Oxidative stress.
Yeah.
That means free radicals, right?
Loads of them. An uncontrolled production of reactive oxygen species, ROS. These things are highly unstable molecules, free radicals, and they just wreak havoc.
Havoc how? What do they attack?
Pretty much everything crucial. They go after cell membranes. That's called lipid peroxidation. They damage proteins, even DNA. It's just widespread molecular damage.
And this damage, this neurotoxicity, leads directly to more neurons dying off, even after the blood flow is back.
Yes, it causes delayed neuronal cell death. And that really determines how severe the long-term outcome of the stroke will be.
And I remember reading the sources mention this damage isn't uniform. Some brain areas get hit harder.
That's a key point. Certain regions are much more vulnerable. The hippocampus, for instance.
It's important for memory.
Critically. And specifically, the CA1 region within the hippocampus. It's known to be exquisitely sensitive to this kind of IR injury. Lots of cell death happens there.
Okay, that sets the stage perfectly for this new study. It's preclinical, using an animal model.
Right. Hypothesis-driven work by Irfani and colleagues.
And they're looking at esketamine as a potential way to protect against this IR damage. Why esketamine specifically?
It comes down to how esketamine works. It's known primarily as a non-competitive NMDA receptor antagonist.
Okay, and how does blocking NMDA receptors help during that reperfusion phase?
Well, during IR, you get this phenomenon called excitotoxicity. Basically, nerve cells get overstimulated to the point where it becomes toxic.
Too much signaling.
Way too much.
Right.
And that overstimulation is partly driven by excessive activation of these NMDA receptors. So the thinking is, if esketamine can dampen down that NMDA activity,
it might reduce the excitotoxicity, reduce the neurotoxicity, and ultimately protect the neurons.
That's the hypothesis. Reduce that damaging cascade right as the blood flow returns.
So how did they test this? What was the setup?
They used a standard model in rats, 28 male Wistar rats. They induced ischemia by temporarily blocking the common carotid arteries.
For how long?
For 20 minutes. Then they removed the blockage to allow reperfusion, mimicking what happens after stroke treatment.
And the esketamine was given after the blockage was removed?
Immediately after, right at the start of reperfusion. This targets the IR injury phase specifically, not the initial ischemia.
Makes sense. And they compared different groups.
Right. 4 groups essential for comparison. You had your sham group, healthy controls, no stroke. Then the IR group. They got the stroke injury, but no treatment. This is your baseline for damage.
Got it. The injury control.
Exactly. Then the 2 treatment groups. One got a low dose of esketamine, 0.1 milligrams per kilogram, injected intraperitoneally, or IP.
And the 4th?
A higher dose group, 0.25 milligrams per kilogram, also IP, right after ischemia. This dose comparison is important.
Yeah, to see if there's a dose-response effect. More drug equals more protection.
Precisely. That strengthens the case if you see it.
So, to see if it worked, they needed to measure the damage and the brain's defenses, specifically in that vulnerable CA1 region of the hippocampus.
Correct. They looked at key molecular markers. For damage, they measured MDA malondialdehyde.
Which tells you.
It's a direct marker of that lipid peroxidation we talked about.
Yeah.
Basically, it shows how much the cell membranes are being damaged by free radicals. High MDA means high oxidative damage.
Okay, that's the damage side. What about the brain's own defense system? How did they measure that?
They looked at the activity levels of the brain's main antioxidant enzymes. These are the good guys, the cleanup crew.
Like SOD and...
SOD, which is superoxide dismutase, also sketuc, catalase, and GPX, glutathione peroxidase. These three are the major players in neutralizing those harmful ROS.
So, ideally, with an effective treatment, you'd want to see MDA levels go down.
Right, less damage.
And the activity of SOD, CAT, and GPX go up.
Exactly. You want to see the brain's defenses getting stronger or being better preserved.
Okay, let's get to the results then. What happened in the IR group, the ones who just had the injury with no treatment?
Well, as expected, it was bad news. Their MDA levels shot way up, confirmed significant oxidative damage in the CA1 region.
And their natural defenses, the enzymes.
Their activity was significantly reduced. SOD, CAT, GPX, all of them took a hit. It shows the natural defenses were just overwhelmed by that sudden oxidative stress during reperfusion.
Okay, so the stage is set. Now, the crucial question, what happened when they gave esketamine? Did it change that picture?
It absolutely did. Esketamine significantly reduced the MDA levels compared to the IR-only group. Less damage.
Was there a difference between the low and high dose?
Yes, a very clear difference. The higher dose, 0.25 milligauge, showed a much more pronounced reduction in MDA. Statistically, it was highly significant, p less than 0.001.
Wow, okay. So it clearly reduced the damage, especially at the higher dose. But did it work by boosting the defenses like you hoped? What about those enzyme levels?
And this is perhaps the most interesting part. Yes, it did. Esketamine significantly increased the activity of all three key antioxidant enzymes, SOD, CAT, and GPX, in that CA1 region.
So it wasn't just passively blocking damage, it was actively helping the brain fight back.
That's what the data suggests. It seems to be bolstering the brain's own antioxidant defense system.
And was that enzyme boost also dose-dependent, better with the higher dose?
Absolutely. Again, the higher 0.25 milli/kg dose led to significantly greater increases in the activity of all three enzymes compared to the low dose. Really clear dose-response relationship there, again, highly statistically significant.
So you've got lower damage markers, higher defense enzyme activity, and both effects are stronger with the higher dose. That paints a pretty convincing picture for the mechanism.
It really does. It strongly suggests that esketamine is protecting the brain, at least in this model, primarily by enhancing its natural ability to cope with the oxidative stress of reperfusion. It's helping the brain help itself.
Okay, let's think about what this means clinically. If this mechanism holds up, how could it potentially change how we approach stroke treatment? You mentioned IR injury is a big barrier to long-term recovery right now.
Right. Think about the timing. Current acute treatments, like getting rid of the clot, have a very narrow time window, often just hours after the stroke starts.
Yeah, time is brain.
But this reperfusion injury, the oxidative stress and cell death it causes, that can actually continue for hours, maybe even days after the blood flow is restored.
So esketamine might offer a way to intervene after the initial clot busting, extending the therapeutic window.
That's the potential implication, yes. If esketamine proves effective as a neuroprotectant against IR injury, you could potentially administer it after a successful thrombectomy or TPA treatment.
To protect the brain tissue that was just saved from that second wave of damage.
Exactly. It wouldn't replace the need to restore blood flow quickly, but it could complement it. It shifts the focus slightly from just opening the vessel to also actively protecting the recovering brain tissue in the hours and days that follow.
That could make a huge difference in long-term outcomes.
It could. It offers a potential strategy to mitigate that damaging second phase that we currently don't have great tools for.
This preclinical study shows esketamine may protect the brain by directly bolstering its antioxidant defenses against reperfusion injury.
This provides a clear mechanism and a strong rationale for more research into using esketamine as a neuroprotective agent for stroke patients, while stressing it is still an animal study.
Definitely needs more research, but promising.
Very promising. And, one final thought for you to consider. If this neuroprotective role is confirmed in more complex models, maybe even human trials eventually, a key question will be the dosage.
Right. Can you get this protective effect at a dose that doesn't cause the significant dissociative or anesthetic effects we see with higher clinical doses of ketamine?
Exactly. Finding that therapeutic sweet spot where it's neuroprotective but still safe and tolerable as an add-on therapy after a stroke, that will be the next big challenge.
Until next time, stay curious.